वेलकम एवरीवन। सो आज जिस टॉपिक के बारे में हम बात करने वाले हैं दैट इज सेल द यूनिट ऑफ लाइफ। नाउ एज वी ऑल नो दैट वी कॉल सेल द फंडामेंटल unit of life, right? Now moving on, as we all know that all living organism, they are made up of cell, right? Whether it is a single cell organism or a multicellular organism, that the basic composition, or we can say the basic structure of a living organism, is a cell. And we know that unicellular organism, they are capable of independent existence. That, for example, if we say in a multicellular organism there are various functions just such as respiration, digestion, excretion, movement. For that each and every function there is a specific organ system which are dedicated to it. But in case of unicellular organism, a single cell is capable of doing all the functions which are necessary for living, right? Now anything less than a complete structure of a cell does not account for a independent living, right? So basic independent living unit, we can say that it is a cell, and below the cell nothing is considered as a living. So हम ये कह सकते हैं that the cell is a fundamental structure and functional unit of all living organism, right? First live cell was described and sought by Anton von Leeuwenhoek, and Robert Brown discovered the nucleus. Now let's just understand what is cell theory. Basically एक German botanist थे Matthias Sheldon. He examined a large number of plants and observed that कि जितने भी plants हैं they are composed of different kind of cell which form the tissue of the plant, right? Now at the same time another British zoologist Theodore Swan, right, he reported that the cell of animal they have a thin outer layer which today we call as the plasma membrane, right? Now Matthews, Sheldon and Schwann Theodore, they proposed the hypothesis that the bodies of animal and plant are composed of cell and products of cell, right? Later another scientist known as Rudolf Virchow, he gave the final cell theory. Now according to this cell theory, we can say that all living organism they are composed of cells and products of cell, and all cells they arise from pre-existing cells, right? So this is the basic two postulates of the cell theory. Now let's just discuss an overview of cell, right? We already know that cells where there is a nuclear membrane, or we can say a membrane bound nuclei, such cells are called as eukaryotic, whereas cells who lack a membrane bound nucleus they are known as prokaryotes. So in basically both prokaryotes and eukaryotes they have a semi fluid matrix which is known as the cytoplasm of the cell. Now eukaryote cell has a distinct feature which is known as the membrane bound organelles such as endoplasmic reticulum, the Golgi complex, lysosome, mitochondria and etc. Whereas prokaryote cells में there is absence of a membrane bound organelles. Now ribosome एक इकलौता non membrane organelle है which is present in all cells, चाहे वो यूकैरियोटिक हो और प्रोकैरियोटिक हो. Within cell, ribosome are not only present in the cytoplasm, but they are also present in two organelles, that is mitochondria and on rough endoplasmic reticulum. Now you can see in this diagram there are various cells from the human body. This is the smooth muscle cell, the blood cell, the nerve cell, male sperm cell and female ovum, as well as fat cell. Now you can see that there are different shapes of these cells according to their structure and function. Now we will discuss the prokaryotic cells. So basically prokaryotic cells, they are generally smaller and multiply at a rapid rate in comparison to that of eukaryotes. They vary greatly in shape and sizes, right? So the four basic shapes of bacteria, they are bacillus, coccus, vibrio and spirillum. So in this diagram you can see that this is the cocci shape, right? The singular we can say it's cocci, then this diplococci means there are two. When it is in a chain like structure, it is known as cephalococci, and the bacillus, we can see that bacillus, it is a rod like structure, right? Now you can see a vibrio, another which is a comma shape, right? You can see the comma shape here, and spirillum means a spiral shaped bacteria. These are the four basic shapes. Now all prokaryote cell, they have an external layer surrounding the cell membrane, which is known as cell wall, and this cell wall is absent in mycoplasma, right? The fluid matrix filling the cell is the cytoplasm, we already know that. Now in case of prokaryotic cell, there is no well defined nucleus, so the genetic material is basically naked and not enveloped by a nuclear membrane. Now in addition to the genomic DNA that the prokaryote cell have, there is a extra circular DNA which is present outside the genomic DNA, which we called as plasmid, right? Nuclear membrane is found in eukaryotes. No organelle like the one in eukaryotes are found in prokaryotes, right? So there are no membrane bound organelles in prokaryotic cell except for राइबोसोम. राइबोसोम प्रोकैरियोट और यूकैरियोट दोनों ही सेल्स में पाया जाता है. A specialized differentiated form of a cell membrane, which we call as mesosome, is found in prokaryotes, and it is also a characteristic function. Let's discuss the cell envelope and its modification. Right, mostly prokaryote cells, particularly the bacteria cell, they have a complex cell envelope. The cell envelope consists of a tightly bound three layer. You can see here that outer glycocalyx, followed by a cell wall, then a plasma membrane, right? So bacteria can be classified into two groups on the basis of differences in their cell envelope, that is gram positive and gram negative, right? You can see here the gram negative cell wall has a thin layer of peptidoglycan, thin peptidoglycan, whereas the gram positive, it has a thick layer of peptidoglycan. Now moving on, a special membranous structure is the mesosome. As you can see in this diagram that this structure is a mesosome, which is basically a extension of plasma membrane into the cell. Now this extension are in the form of vesicle, tubules and lamella. If motile, the prokaryote cells, especially bacteria cells, they have flagella, thin fibrous extensions are arising from their cell wall, right? So bacteria show a range of flagella in cases in number of in case of number and arrangement of flagella. So bacterium flagella, as you can see in this diagram, is basically composed of three parts, right? First is the filament, then we say a hook and a basal body. So basically the filament is the longest portion which extends from the cell wall surface to the outside, right? So beside flagella, pili and fimbria are also surface structure of bacteria, but they do not play a major role in motility. Ribosome is the only cell organelle which is found in the prokaryotic cell. Ribosomes are generally about 15 to 20 nanometer in size. They made up of two units, right? 50s and 30s, and when they present together they are formed 70s prokaryotic ribosome. So in this diagram you can see that there are the two subunits. Right, this one is a larger unit and it is the smaller unit. Inclusion bodies are basically the reserve material in prokaryotic cell, right, that are stored in the cytoplasm in form of inclusion bodies. They are not bound by any membrane system, hence they lie free in the cytoplasm. Now let's just discuss eukaryotic cell. In eukaryote there is a extensive compartmentalization of cytoplasm through the presence of membrane bound organelles, right? So eukaryotic cells, they possess an organized nucleus with a nucleus envelope, or we can say a nucleus membrane. Now in addition to that they have a complex locomotory and cytoskeleton structure which we will discuss later. The genetic material in case of prokaryotic cell, we have studied that it is not in a membrane structure but it is loosely found in the cytoplasm, or we say there is a naked genetic material present in the prokaryotic cell, right? But when we talk about eukaryotic cell, so in eukaryotic cell the genetic material is basically organized into chromosome, right? And chromosome is a dense form of genetic material. So all eukaryotic cell, they are basically not identical. Plant and animals are the example of eukaryotic cells, right? Of animal cell and a plant cell, right? Now in plant cell we have cell wall, right? And animal cell has no cell wall. Plant cell has chloroplast, but in case of animal cell there is no chloroplast. Plant cell have large central vacuole, where animal have numerous or no vacuoles, right? Now moving on, let's just discuss cell membrane. So cell membrane is mainly composed of lipids and proteins. Now the major lipids are phospholipids, which are arranged in a manner of a bilayer. So the lipids of cell membrane, they are arranged in such a way that the polar head is towards the outer side and the hydrophobic tail towards the inner part. This ensures that the non-polar tail of saturated hydrocarbon is protected from the aqueous environment, right? In addition to phospholipids, cell membrane also contains cholesterol, proteins and carbohydrates. So the percentage of protein to lipids in case of an human cell, that is 52% of protein and 40% of lipid, right? Now the proteins, they are also classified as integral and peripheral. So the proteins which lie on the surface are known as peripheral, whereas the proteins which are, we can say, partially or totally buried in the membrane is known as integral protein. Now this is the diagram of a cell membrane, or we can say a fluid model of cell membrane. Right, so we can see that this is the lipid bilayer. So let's just magnify this diagram. So this is the lipid bilayer, right? So as we have learned that the polar head towards the outer and the hydrophobic tail towards the inner part, right? So this region is the polar head and this region is the hydrophobic tail, right? We can see that there are carbohydrates also present on the cell membrane. Now when we talk about proteins in this diagram, we can see that there are such peripheral proteins which are present on the surface, whereas some of the proteins, they are embedded fully or partially, right? Here we can see that this is an integral protein, this is also an integral protein, as it is fully embedded in the cell membrane, right? Approved model of the cell membrane was proposed by Singer and Nicholson, widely accepted as a fluid mosaic model, which we have discussed here. Now according to this model, the quasi-fluid nature of lipid, it allows lateral movement of protein within the overall bilayer, right? And the fluid membrane is important for functions like cell growth, intercellular junctions, secretion, endocytosis and cell division. The most important function of plasma membrane is the transport of molecules across it, and we know that the plasma membrane is semi-permeable membrane, right? It allows only certain objects, or we can say a certain molecules, to pass through it, right? Now moving on, solutes, they may move across the membrane by the process which is known as simple diffusion, and this process of simple diffusion, it works along the concentration gradient, right? That is from higher concentration to the lower, right? Water may also move across this membrane, with the concentration gradient, right? Now movement of water molecules by diffusion we called as osmosis, right? Now in case of polar molecules, they are not able to pass through the non-polar lipid bilayer, so they require a carrier protein to facilitate this transportation across the membrane, right? Molecules against their concentration gradient. Such transport requires an energy, and energy in the form of ATP. It utilizes, and this process is called as active transport. For example, the sodium potassium pump. Now let's just discuss cell wall. So basically cell wall is a non-living rigid structure and forms an outer covering for the plasma membrane of fungi and plants. Now cell wall does not only give the shape, it also protects from mechanical damage and infection, but helps in cell to cell interaction and barrier to undesirable macromolecules, right? So different cell have different composition of their cell wall. So when we talk about algae, they have a cell wall made up of cellulose, galactans, mannans and mineral like calcium carbonate, while other plant it consists of cellulose, hemicellulose, pectin and proteins. Now cell wall of a young plant, or we can say the primary wall, is capable of growth which gradually diminish as the cell mature and the secondary wall is formed. Now the middle layer, mainly composed of calcium pectate, which holds or glues the different neighboring cell together. The cell wall and the middle lamella may be transferred by plasmodesmata, which connect the cytoplasm of neighboring cell. Now we'll discuss the various cell organelles present in a cell. Now first of all we'll discuss endoplasmic reticulum. So endoplasmic reticulum basically is divided into two types, that is smooth endoplasmic reticulum and rough endoplasmic reticulum. So when we talk about structure, smooth endoplasmic reticulum, there is an absence of ribosome on its surface. Now in this diagram you can see that this is the smooth endoplasmic reticulum and there is no presence of ribosome on its surface, whereas in case of rough endoplasmic reticulum we can see these minute structures which we called as ribosomes. Now when we talk about function, smooth endoplasmic reticulum, they synthesize lipids, steroids and hormone, whereas rough endoplasmic reticulum, they synthesize protein for secretion, membrane insertion and organelles. Now we have already discussed that smooth endoplasmic reticulum has no ribosome, whereas rough endoplasmic have a ribosome attached to their cytoplasmic, right? Now smooth endoplasmic reticulum, they play a significant role in detoxification of drugs, whereas rough endoplasmic reticulum has no role in detoxification. Smooth endoplasmic reticulum is involved in the carbohydrate metabolism and lipid metabolism, whereas rough endoplasmic reticulum is focused only on protein metabolism, right? The smooth endoplasmic reticulum is abundantly found in liver and adrenal gland, whereas rough endoplasmic reticulum is abundantly found in pancreatic cells. Now from the microscopic view, smooth endoplasmic reticulum appeared a smooth and a tubular structure, whereas rough endoplasmic reticulum is observed as a rough and flattened due to the presence of ribosome. Golgi apparatus is basically named after a scientist called Camillo Golgi who discovered it, and he observed a densely stained reticular structure near the nucleus. Now in this diagram we can see our Golgi body or Golgi apparatus is present close to the nucleus. Now it consists of a flat disc shape sacs. We can see they are flat, right, a shape of a disc, and which are parallel to each other, and there are two faces in case of Golgi apparatus, a distinct convex face, or we can say trans face, and a concave cis face, right. So Golgi apparatus performs the function of packaging material that is to be delivered either within the cell or outside the cell, right. Materials to be packed in the form of vesicles from endoplasmic reticulum. They fuse with the cis phase and move towards the maturing phase. So in this diagram we can see the packed material. They will move from the cis phase to the mature trans phase, and we can see that there are various secretory vesicles, right. Now a number of proteins synthesized by ribosome on endoplasmic reticulum are modified in the system of the Golgi apparatus before they are to be released. Now Golgi apparatus is an important site for the formation of glycoproteins and glycolipids. This is all about today. If you like this video, don't forget to like and subscribe. And we are starting the crash course batches for class 11th and 12th. If you want to join the batch, the link is in the description box.